Hi, my name is Claire Demarque and I'm a postdoc in Ryoaki Matsunami's lab at Duke University. Today, I'm here to present you my talk, which is about how an enhanced evolutionary capacitance can allow olfactory receptor with cryptic mutations to be functionally expressed. But first, I would like to do a quick reminder on how we smell odors. Odors are composed of volatile molecules, which are very diverse and are emitted by odorant sources. When you breathe, these odorant molecules enter into your nose to reach your nasal cavity, which is just behind your eyes. Your nasal cavity is covered by the olfactory mucus, which prevents the olfactory mucosa from drying. This layer of water is a barrier for the hydrophobic odorant molecules. In the olfactory mucus, there is proteins called odorant binding proteins that have the property of having an hydrophobic cavity and an hydrophilic surface. So they can bind odorant molecule into the cavity and transport the odorant molecules into the mucus. The odorant molecule can then reach the olfactory sensory neurons that are here represented in red, which projects cilia into the mucus. Let's have a closer look to the membrane of this cilia. The olfactory receptors that are here represented in yellow are embedded into the membrane. Olfactory receptors are GPCRs, so they share the same structural features, meaning 7 transmembrane helices. The odorant can or cannot have an affinity with the receptor, but if it is the case, the odorant binds into the cavity of the olfactory receptors, which triggers the activation of the receptor, which switches from an inactive state to an active state. And the end of this cascade of reaction is the opening of an ion channel, which allows ions from the exterior part of the neuron to go inside the neuron, creating the depolarization of the membrane. This is here a very important step, because the chemical signal from the odorant molecule just have been transformed into an electric influx. So we saw now that olfactory receptors are the first step in the detection of smell. Interestingly, these olfactory receptors are also shown to be expressed outside of the olfactory systems, in brain, in skin, in gas, or even spermatozoid. The exact function of olfactory receptor outside of the olfactory system is not very much known, but we can suspect that the role of olfactory receptor is bigger than only allowing us to smell thousands and thousands of odors. Olfactory receptors are part of the G protein-coupled receptor family. This family is composed by many types of receptors, going from the chemokine receptors to the adenosine receptors, and include the olfactory receptor, which are a very diverse with numerous member subfamily of the GPCRs. In fact, olfactory receptor actually represent more than 50% of the rhodopsin-like subfamily of GPCRs. So how does GPCRs function? First, they have to be expressed at the membrane of a cell. Second, they have to bind a group of ligand which is specific to this receptor. Third, upon the binding to a ligand, this receptor have to switch from inactive to active states to trigger the signalization pathway of the cell. All GPCR, including olfactory receptors, are able to fill these functions in their natural environment. However, olfactory receptors are really not performing well in heterogeneous cells, meaning that they are not expressed in a cell model. We can measure the cell surface expression of an olfactory receptor thanks to flow cytometry. Olfactory receptors are expressed in cell model, here Hanna-Tree cells, and they are raw tagged. That allows us to attach an antibody to this raw tag and then a fluorescent target to monitor the fluorescence at the surface of the cell and evaluate the cell surface expression of the receptor. When we monitor the cell surface expression of two olfactory receptors, OLFR 539 and OLFR 541, and we compare this expression with the GPCR, the muscarinic tree receptor, we can see that the level of expression is highly lower for olfactory receptors. And it is even more dramatic when you consider that OLFR 539 is one of the most expressed olfactory receptors. However, this low expression is sometimes enough to evaluate the functionality of the receptor, and we were able to see the functionality of some receptor through the luciferase assay in Hanna-Tree cells. But this low expression is really blocking the advance that could be done on our understanding of the olfactory receptor function. This lack of expression in cell model prevents a detailed study of their function, the determination of the OR structure, a precise description of the olfactory receptor-odorant interaction. So why olfactory receptors are so poorly expressed in heterologous cells? How should we interpret this characteristic, which is very specific to the olfactory receptor family in the big family of the GPCRs? First clue. In 2004, Hiroaki Matsunami's lab discovered that chaperones called RTP, receptor transport protein, are helping the expression of olfactory receptor in heterologous cells. In fact, as you can see here, cells are transfected with olfactory receptor. The olfactory receptor cell surface expression can be noticed in white in those pictures. When the RTPs are co-expressed with the olfactory receptor, finally, the receptor is addressed to the surface of the cells. The hypothesis is that RTPs are helping the olfactory receptor to fold in the endoplasmic reticulum and then being transported through a vesicle to the plasma membrane. The second clue is that stability seems to be the key for the olfactory receptor to be expressed at the surface of the cell without the help of any chaperones. We now come back to the example of OLFR 539. And we found that one single mutation at the position 154 is able to decrease dramatically the cell surface expression of this receptor. So we were very intrigued by this single mutation, which is able to totally kill the cell surface expression of this receptor. To understand better this mechanism, we used in silico tool and notably molecular dynamic simulations to see the behavior of both of these receptors in the model of a membrane. When we run the molecular dynamic simulations with our OLFR 539, we can see that this receptor is forming a clean bundle into the membrane model. However, when we start the same molecular dynamic simulations with the same receptor and only the position 154 mutated, we can see that the results are dramatically different. In fact, now the receptor is no more forming a bundle of a GPCR. The helices are broken and start to unfold. We run several of these molecular dynamic simulations on different systems, and we can make the point that the stability in silico of the receptor is very much linked with the experimental cell surface expression of olfactory receptors. So to resume, olfactory receptors are of an exceptional diversity and with very numerous number in the GPCR family. A lot of them show a lack of stability in the membrane, and the same ones are in need of chaperones to be expressed, and it is true for heterogeneous cells, and it is also true for olfactory sensory neurons. It does remind the case of proteins that had to have mutations to adapt during evolution, but those mutations came with a price, the price of a lack of folding and expression. Those mutations that helps proteins to survive during evolution and adapt to their environments are called cryptic mutations. This is when evolutionary capacitor can intervene. Evolutionary capacitors are other proteins, sometimes chaperones, that helps these proteins to compensate the effect of these cryptic mutations. This is the case of HSP90 protein, which is a chaperone, which is helping a lot of different proteins to fold properly. So if this hypothesis is true, that means that the ancestor olfactory receptors were very much functional without the help of any other proteins, meaning without the help of any evolutionary capacitor that could be the RTPs. We then need to build olfactory receptors that are as close as possible as an ancestor olfactory receptors. And we also need to try to improve their stability in membranes to see if that can help their cell surface expression. One single strategy can address both of these points. The consensus strategy. The consensus strategy basically says that in a family of proteins, the amino acid the most conserved at a certain position is more contributing to the function of these proteins than a random amino acid. This strategy has never been used in GPCRs because regular GPCR only possess a few members. But olfactory receptors have these very specific features to be very numerous. We apply this consensus strategy to subfamily of olfactory receptors. And I'm going to first show the OR10 family. You can see here the alignment of all OR wild type from the subfamily number 10 in human. The building of the consensus sequence was made by looking at each amino acid was the most conserved for each position. Here I'm just showing a part of the alignment, but we applied this to all 300 positions of the sequence of these ORs. We then obtain a receptor which is called a consensus receptor, which doesn't exist in nature. So as those receptors are new, we needed to see if they were well expressed in the cell surface of cell models. if they were able to answer to a group of odorants. So if they were acting as any olfactory receptor, since they already possess all the amino acid hallmarks of the olfactory receptor family. So we first tested the cell surface expression of all the members of the OR10 human family. And you can see here that most of these receptors are actually not showing a high cell surface expression. We can see that OR10 consensus is way more expressed than any wild-type receptor observed so far. This success encouraged us to continue with other subfamily of olfactory receptors. And we applied the same strategy on 9 subfamily of olfactory receptors. And you can see here that for 6 of them, the same effect was observed, where we obtained a consensus olfactory receptor, which is way more expressed than any native olfactory receptors. And if you now compare this expression with the one of a GPCR, the muscarinic receptor, you can see that we tend to reach the same level of expression of a non-olfactory GPCR, which never happened for an olfactory receptor so far. Now that we know we have very well expressed olfactory receptor, we needed to know if those receptors were still functional. We realized a large screening of odorant stimulation on all these receptors, and we found that some of them were able to respond to odorant molecules. Those receptors were very much acting like any odorant receptors, meaning that they were binding a specific group of odorants depending on their category. Now that we have those great models of olfactory receptor, we wanted to see if stabilization strategy can help their cell surface expression. To test stabilization strategy, we chose to continue with OR10 and OR52 because they were showing already great expression in our cell model, and they were also able to answer to odorants. In collaboration with Vaidhi Nagarajan and Sumali Ghosh, we applied stabilization strategies that are very common in the GPCR world. This strategy consists to insert ionic lock in the structure of the receptor at locations that are not involved in ligand binding or activation process. The insertion of these ionic locks will rigidify the structure and make the structure of the receptor more stable in the membrane. So here we choose to insert these ionic locks in between the helix 8 and the intracellular loop 1. We inserted negatively charged and positively charged residue in this location, creating those ionic interactions, locking the receptor in this part. We first measured the cell surface expression of those new mutants, and as you can see here, the cell surface expression of the mutants is even higher than the original consensus receptors. And now, when you compare with the muscarinic receptor, we actually overpassed our expectation and we obtained olfactory receptors that are more expressed than a non-olfactory GPCR that already have been crystallized. Of course, we wanted to be sure that this rigidity inserted in the structure doesn't prevent the activation mechanism of the receptor. So we tested those receptors also in luciferase assay. As you can see, the receptors are still very much responding to the same odorant than the original consensus receptor in those responses. So we confirm that olfactory receptors that are close to ancestor olfactory receptors are actually functional without the help of any chaperones. So starting from these consensus ancestor receptors, they had to have cryptic mutation to be able to diversify. But those cryptic mutations were coming at the price that those receptors were no more functional or able to fold to reach the cell surface of the olfactory sensory neurons. And this is where evolutionary capacitors like RTPs and surely others were intervening to help those receptors to be addressed at the surface. Thanks to this mechanism, we are now able to spend thousands of orders thanks to a repertoire of olfactory receptors constituted by hundreds of them. These results are very important for many fields, and notably basic science, because now we have OR models that allow us to push the limits of what we could do so far. And all the knowledge gained on these OR models could be applied to any wild-type olfactory receptor since they are representing subfamilies of native olfactory receptors. Since those receptors are also behaving very well in cells, this receptor could be used for electronic nose or any other industrial application which will consist to detect odorant molecules. And now I want to thank my lab, Hiroaki Matsunami's lab. I also want to thank warmly my collaborators, and I also want to thank the NIH, which is providing me funding to continue my experiment. And I also want to take a few seconds to introduce you to my YouTube channel, which is called Who Knows, and which is a very newborn YouTube channel. The aim of this YouTube channel is to spread the science of others to the world. And to finish, I want to thank you for listening to my talk, and I wish you a happy virtual ISA 2020.